Is Professor Marcin Moniuszko, and the talk will be morbid study in research and omics, markers of personalized diagnosis and treatment of non-small cell lung cancer.
Thank you. Good afternoon, and I would like to, in the beginning, to thank the organizers for inviting me to this really exciting event, as I can see. And from the very beginning, I would like to explain I'm not going to talk about the results because we are not having results yet. However, I would like to talk about some kind of strategy and some kind of strategic approach that we have started at Medical University of Białystok just in order to study in general a number of civilization diseases. In this specific talk I will be talking about the non-small cell lung cancers. So I think that like 10 years ago we've made a strategic decision at our university that we were not going to only talk about personalized medicine, but we will try to do something about it, really do something. So as we all know, last decade was quite generous in terms of funding, so we were also lucky to invest in funding of our laboratories, of our clinical centers, experimental medicine and so on and so on, but it was all done for purpose. So our goal was actually not just to invest it, keep it, and do some separate projects and let's see what's going to happen. From the very beginning, our investments were actually kind of focused on doing something really big, something that will be joining different teams and different experts, and something that really will result in some kind of diagnostic and therapeutical solutions. And the project I will be talking today now is one of these examples, and we kind of picked this project as our kind of flagship enterprise, since this is something that actually is the example of really joint forces that can make a difference. And the MOBIT project, which is actually the acronym of the Molecular Biomarkers for Individualized Therapy of Non-Small Cell Lung Cancer, is the example of this kind of joint actions. And in these actions, Medical University of Białystok is really honored to be a leader of this project. However, we do have to really, and this project wouldn't be possible if we were not in collaboration with our renowned experts. One of these experts is sitting with us in the audience. This is Professor Monika Kaczmarek. We are lucky to collaborate with her, with the Polish Academy of Sciences. She is one of the few experts in proteomics in our country, so we are really, as I said, lucky to stay in touch with her, but also we do have some clinical collaborates, the Poznań Medical University of Poznań, also the regional hospital of Olsztyn, and several groups from Germany as well, the Heidelberg University, and the Indivimed. Indivimed is the company that actually has set up the really highly standardized SOPs for collection. And I will mention about this because this is something that, unfortunately, also it's being changed. It kind of, in the course of time, this attitude is changing, but still not many people really appreciate not only the value, but just the critical necessity of really taking care of the good biobanking samples. This is something that we forget about too often. So that's the team, and what's the goal of the team? So the goal of the team and the rationale is that we all know we don't have to repeat it, but there's no one cancer. Even if we take a look at one histological group of cancers, or we still see a lot of heterogeneity. So this is something that has to be really taken into account much more in the deep. And of course, molecular patterns, not just the single genes, not just the even sets of single genes, but just what we call right now genomic analysis is something that has to be done sooner or later. So this is not the matter of whether we like it or not, but that has to actually be done. And based on that, especially in this kind of diseases, as we know, non-small cell lung cancer has really very, very low rates of progression-free survival, really low rates of responses to standard chemotherapy. And of course, the 5-year survival is just dramatically low. So this is, and of course, on the other hand, this is one of the most frequent diseases, malignancies in our country. So there was no, I think, question to explain why did we focus on this particular cancer. This is one of the, unfortunately, most dangerous killers in our society. And our goal was to focus and to integrate several approaches. So we started from the very beginning. So we started from really something that seems to be trivial, but it's not trivial if we look at several situations that are showing us how critical can be if we don't care about biobanking. So we started with the good standard operating procedures for collecting samples. And this is actually the very important part of the project. And it's, as I will say, it's not about buying new freezers. It's not about, buying the automated system that will allow us the, identification of samples. It's not about the liquid nitrogen and the most modern liquid nitrogen. It's everything about philosophy, about the strategy, about making people to start to believe that biobanking, that collection, the really appropriate collection of samples is something extremely critical. Because as I will show you some examples, if we don't look at this, then we have no warranty that our data can be reproduced at any time. And this is actually one of the basic reasons for which, if we look at several analyses coming from, for example, United States, they recently came up with this analysis showing that 70% of the data that have been actually funded by NIH, by different grants, cannot be reproduced. And one of the main reasons, not just the scientists are trying to, are liars. It's not true. It's maybe some, yes, but let's assume that it's not us, and the majority of them are the honest people. But majority, and the West, one of the biggest problems is this, that we never know on what kind of quality of data we're working on. So the appropriate biobanking is something that is extremely critical. And we've made this effort. It costed us a lot. It costed us to really, not only money, it costed us to really convince people. I wouldn't say convince, to convert people to this. Because people have to start to believe in the significance of that, in the importance. If they don't care. And in majority of places all around Poland, I believe, and Europe is not the exception for this, people, they just don't care. I mean, the sample has been collected. What's going to happen further, we'll see. Sometimes it's a good, let's say, pathway. Sometimes it just, put it somewhere, and people don't know what they are comparing to. So that was something that we've done in the very beginning. Of course, once we have the good quality samples, so next we decided that we will not be focusing on just doing, several, I would say, very specific analysis. We decided that we have to take a step back. We don't have the theory. We don't have hypotheses. Of course, we do have plenty of hypotheses in our minds, but this hypothesis shouldn't actually stop us from doing everything. They shouldn't stop us from finding something new. So we decided that we will do, I would say, basically everything, in everything. By that, I understand that we decided, for example, for those patients that they have the tumor that can be resected, that are still eligible for the surgery. So we don't just take the material from the tumor. We take the material from different parts of tumor. So the tumor center, the tumor margin, the different, we assess the surroundings of the tumor, and everything is being analyzed. And once we take this, we also collect the material from, of course, the blood, and from blood we separate serum, plasma, PBMCs. We also take urine. We also take, if possible, some aspirates if they can be collected in several patients. So we look at this in a very comprehensively. So we don't think that some material is better or worse. No, we just take everything. And by what I understand by saying that we analyze everything. So the quantity of material, of biospecimen material we can collect, allow us really to really do a lot of stuff. So far, and I will show you, so we are just doing any kind of sequencing, including whole genome sequencing, not just whole exome sequencing, whole genome sequencing, transcriptomics, RNA-seq, and microRNA-seq, but also proteomics, metabolomics, radiomics, and so on. I will come up with this in a moment. So all this data, it produce just this kind of amount of data that cannot be just analyzed by simple statistics. So it all gets to it's actually the very ideal tool for developing bioinformatic algorithm solutions. And this is something that's actually the platform for creating this kind of solutions. And the development of such platform is another step of this project. And this is something when we are at this moment, that we are developing this platform that at the end will allow us to create the reference model for personalized diagnosis and therapy. So when we look at this, it's nothing strange. It's nothing exceptional when we look at this. However, the problem is that if we look separately at all these points, they are just seem to be trivial. But unfortunately, in what's not happening on the standard on a daily basis, not only in Poland but in Europe, that very few centers, if any, they're trying to implement this kind of very comprehensive approach to this kind of science. We wouldn't have done it if we didn't have the grant. And this grant is coming from, of course, from the Polish NCBR. But by implementing this kind of procedures, we are planning to try to use this kind of approach in also studying over diseases, over malignancies. So this is how it looks like from in this pyramid way of presenting, when we start with biobanking, when we start to collect the data, when we start to analyze, to do analysis, genomic, proteomic, metabolomic, radiomic analysis, clinical data, of course, this is very crucial part. This data would mean nothing if they were not correlated very closely with any kind of clinical data, with the history of the patient, with the comorbidities, with the drug responsiveness, with the drug susceptibility, with the history of the progression-free survival, with the overall survival, and so on and so on. So it all leads to the constitution, this platform, and this platform then, when it's being confronted with some other databases, with some clinical questions, then it can lead to developing of this bioinformatic, let's say, artificial intelligence tools that will allow us to create these decision support tools. Not yet now, but we hope to actually come up with them very, very, very soon. So, biobanking. Biobanking is something that actually, as I said, we also, before that project, we had our theory of biobanking procedures. However, we started to, we thought that we should start to learn from the best, from these institutions that actually earn money on biobanking, so they have to really keep very, very strict standard operating procedures. So in our case, we kind of invited the development company to share the SOPs, and we really actually even what we've done in our hands, we even make them even sharper, just really to be 200% sure that our data, that our biospecimens are being collected in a very, very standardized way. So, as I said, it's not about that we've bought modern freezers and so on, and liquid nitrogen is just better than some other institutions. No, it's not about this. It's about people. Biobanking is about people who understand why they are doing so. So, especially because systems can be done in a very similar way otherwise. But everyone has to understand how important is this. Not only people and biobank. It has to be also doctors, clinicians, nurses, pathologists, and all the people that are really, and they should work together. So they really should understand the value of this, the critical value of the collaboration in the biobanking. So as I said, it's not about equipment, it's about thinking, it's about strategy, it's about philosophy of these people. How important is this? Even if nothing happens, just collection of the tumor, and that has been actually already evaluated by many studies. Just collection the tumor from the tumor leads to the decrease of something like 20% of genes of the entire genome in the tumor. So not mentioning the fact that the longer this tumor sample is lying somewhere or is being subject to some kind of changes of temperatures or some over stimuli, so we don't actually, after some time, we don't know what we are testing. And of course, if we don't look at this, then we can see that that's biology. But in order to be sure that we are really assessing some changes, some alterations, some patterns, in order to be sure that it's not artifacts, let's be honest, that they're not artifacts, we really have to focus most on the quality of biobanking. I know I'm kind of boring talking about this, but this is something that really is really critical. So this system finally it works. It wasn't easy. It wasn't easy to convince people really that it should work like this, but finally it works. And we're going to use it not only for this project, but for all the projects that are being right now conducted, and they will be conducted at our university. So it's also about the quality. If something doesn't work, it's not going to biobank. If something doesn't, let's say, is being checked, double checked, and if something is not okay, it's better to not to have the sample in the biobank, whether to have it and not to be sure whether it's appropriate or not. So that's the philosophy. It's not, we strongly believe in quality than the quantity. It's not about increasing the numbers of patients, number of samples. It's about the quality of the samples, because the data will be more credible if we are more credible about the quality rather than the quantity of this data. And later on, what we are doing, we are in the process of this. As I said, we perform all kinds of omics analysis, starting with, this is molecular biology session, so of course starting with all the whole genome sequencing, with transcriptomic, also the exome analysis, also the epigenomics and RNA sex and small RNA sex. So definitely we'll take a look at the RGNase one as well, and we'll take a look how is it correlated to some other omic changes within the profile. Why are we doing so? I mean, there's no question that, just looking at the genome is something that because we have to look at the genome of the cancer. Cancer is the disease, the genome of the epigenome of many other pathways. But we also have very practical recent proofs of that looking at the genome is not just is fulfilling the scientific curiosity. That can have the very practical actual uses. And one of these uses, for example, right now is, as you probably know, right now we are in the era of the immune checkpoint inhibitors that really are winning the field of the melanoma, and right now they will have been successfully introduced to the treatment of lung cancer. Of course, immune checkpoint inhibitors are not the cure. We don't have the cure for cancer yet, but and there's not going to be something like a cure. Let's face it, that's going to be like several cures, let's say, for different cancers. But immune checkpoints just they really significantly change the rates of survival, and this is something that really has happened. Of course, not in all patients. So there is a question: how can we actually delineate this group of patients that can really be susceptible in a positive meaning of this way to the treatment of immune checkpoint inhibitors? We have different, of course, right now attitudes. The most, let's say, convincing, I mean, the most practical right now is the analysis of PD-L1 expression on the tumor tissue. But however, it has several setbacks that are widely known. However, the last couple of years, the last 2 years actually, have shown us that independently of the expression of the PD-L1 on the tumor tissue, that sometimes can be subject to many alterations, the analysis of mutational burden of the tumor, of the lung tumor, can really, independently of any other factors, can really predict the good responsiveness to the immune checkpoint inhibitors. This is one of the very first studies that was showing this. And this study has been followed by by many other studies. A majority of them have shown the really strong significance and validity of this kind of model. So this is when we assess the mutational burden in the lung tumor. However, if we look at the liquid biopsy, of course, right now everybody is crazy about liquid biopsy, and everyone is having the patent for liquid biopsy, but we don't have still too much data looking at the correlation between liquid biopsy and the tumor itself. So this is something that our study will be also looking at very carefully. Since we have in our hands a very unique set of data, we will see even what kind of genomic profile is present, let's say, in different parts of tumor, and we will correlate this to the different compartments, let's say, the plasma, serum, and so on, urine as well. So I think that our study will be very important part of this discussion in implementing new evidences for the correlations between what really liquid biopsy is showing. Because so far we can see that if we look at the tumor mutational burden liquid biopsy, we still can make a difference, but still we are seeing differences, but we are not seeing something that would, that's scientifically, it can be kind of justified, but we are looking for something stronger to really be sure what kind of changes in liquid biopsy can tell us which patients actually can respond well to some kind of therapy. So I know it's molecular biology session, but I would like to mention about something that we are not just finishing at molecular biology level. We know the genes and of course the epigenomic is just the beginning. Then we have proteins, then we have metabolites, then we have the patient himself or herself. So we analyze metabolomics, we analyze proteomics, this is Professor Monika Kaczmarek, we analyze radiomics, and I will mention a word about this as well, and so many others, especially the clinic. So we look at many other aspects. So for example, we correlate metabolomic profiles with proteomic profiles. We are going to correlate them. We are in the beginning and we are looking forward to looking at this data. I will tell you, I don't have a slide for this, but for example, we were able to see, based on the metabolomic analysis, how closely it was correlated with then what we can see in imaging. For example, imaging with PET-MR, so when we have the uptake of radionuclide by the active tissue of the tumor, we've seen that it was very closely correlated with the metabolomic profile. So it's showing that metabolomics is, again, it's not only about finding biomarkers, it's showing that it can sometimes represent the activity of the tumor. Because we have to remember that not always the tumor is accessible. That's accessible if we have the kind of the early stages of lung tumor. But what about the more advanced stages, when we cannot do a surgery, when the aspiration of the material is a challenge? So we have to have something in the compartments like the peripheral blood that will allow us to understand what's going on, that we can, for example, adjust our therapy based on this kind of discussion. So this PET-MR is something that we're really proud of. This is one of the very few installments in our country. I think there are two, maybe the third one is ongoing right now. We are doing this on the top of the PET-CT because PET-CT is something that's the standard one. So we are not skipping this, of course. These patients are being diagnosed and treated according to all the procedures. But on the top of that, we are doing PET-MR, because by doing PET-MR, we can see the metabolic activity of the tumor. By the way, on the other hand, sometimes PET-MR allowed us to change the diagnosis, because PET-CT was not showing not all the compartments were showing in the right way. This is more like the radiologic and let's say the nuclear medicine discussion. I wouldn't like to go with this, but it's not about again, fulfilling curiosity. Sometimes this kind of examination was changing let's say, the outcome of the patient by kind of enriching the diagnosis. So once we collect all this data, this all huge amounts of different kind of data, clinical data, genomic data, microRNA, transcriptomic, metabolomic, proteomics, radiomic data, other imaging data, and the history of the patient, then it is going to this platform. We called this platform Smart Biobase. It's going to be smart. It's a huge platform, even if we are not talking about thousands of patients. Because I should mention, what we are having here is a couple of hundreds of patients, not thousands of patients. We are not Shanghai Lung Hospital. When we had a visit there, they have like, a couple of hundreds lung surgeries every month. So we are not that. Luckily, we don't have so many lung cancer, but still it's a huge problem in Poland, relatively, if we compare with that kind of country like China. However, anyway, even if it's not Chinese proportions, but still it's a huge database, because the quantity, the number of data, it's just unprecedented one. Because even all this omics analysis, when they are put together, all this radiomics data, we are talking about, I don't want to say that it's not terabytes, it's probably something that is really, really huge. So we don't even have servers for this. We have to put them in the cloud, because we can't build such a server that can really manage to store all this data. But it's just the beginning. It's a beautiful beginning. It's just the beginning, because then this data has to be confronted with clinical questions, that should put them into the practice. With some other databases, the Cancer Genome Atlas, with some other genomic databases, with something that is already known, the driving mutations, and many others. So this is when. At this moment, we start the intellectual work, when we will be trying to find what's the best in this data. And of course, this is the work for the next decades, actually, to come. And we are trying to put in what way we can actually use this data for the benefit of patients and some others. So because the final goal of this project is to come up with solutions, with some kind of diagnostic and therapeutic solutions. So everything what is done here always has to be put against the outcome of the patients, because we, of course, it will be the huge advancement of this project. It will be the finding some new pathways, and that will be, of course, very, very exciting. But at the end of the day, we would like to come up with something that is really come with benefit to the patients, for the future patients. Maybe not these ones yet, because some of them probably will not, unfortunately, survive till the end of this project. But that's our final goal. So we will have to come up with these decision support tools. However, work for this. And this is the work for bioinformaticians. This is the work for people who will be collaborating with clinicians, because it cannot be just done, it has to be done in some kind of directed way. So bioinformaticians, if they are not being directed by doctors, that they will tell, let's look at this, let's say, pathway, what's going to happen to this patient if that kind of sets of mutations we are having. So this is something that will take a lot of time, but this is something also that we are most anxiously looking forward to see what's going to happen. And it's not going to be like one solution, one diagnostic algorithm, or one therapeutic algorithm. I think we're going to have many of them, and this is the work actually for many many specialists, and actually everybody is invited if interested in this kind of approach. So we just would like to come from smart biobase. That is something that is just building the smart biobase wasn't kind of a challenge, but after that time we have to come with building the tools, the artificial intelligence tool that will help to make the better diagnosis, the better treatment for the individualized patients. As for acknowledgements, I would like to put the group of people who are actually most involved in this project. Our ex-rector, past rector, Professor Jacek Nikliński is the head of this project, and again, I have to, she will be angry with me. Professor Monika Kaczmarek is here as well from Polish Academy of Science. This is the event run by Polish Academy of Sciences, so I have to acknowledge Polish Academy of Science all the time, as many times as possible, so I'm doing it right now. There are many people, it's a very joint forces effort, and we are trying to it's not an easy one. There are a lot of troubles on the way, because this is something that no one earlier has done something like this. Not in terms of the intellectual even effort, but in terms of the logistical effort. This is something that's really challenging. So these people are actually they're very happy now. But as I said in the beginning, we are not just we don't just want to finish this project and get it done and forget about this. This is just the beginning. This is just the warm-up. Our strategy is just to implement this kind of approach to any kind of disease that really is posing a problem. And of course, malignancies are those diseases we really owe as a scientific society, as a clinical society, to do as much as possible. So integration of omic data with the detailed clinical history, with radiomic data, is something that we strongly believe that should be done. It's expensive, by the way. So we don't have money for everything, but we do everything to get the money. If somebody in the room has this money, Professor Młynarski always has a lot of money, so we are always inviting him to collaborate for this kind of data, because we can do it also for diabetes, for some other hematological disease, and many others. So you are more than welcome. And of course, we are doing, what are we doing right now when we are using this approach? For example, in our cohort studies, we desperately miss cohort studies in our country that could come up with new molecular, let's say, biomarkers of, not predictive, but prognostic biomarkers, because that's what we are telling. In our project, it's more about prediction of the response to the therapy. But here, how about prognostic biomarkers that will tell what's our, let's say, what are the odds of developing of several diseases. So this is something that we really miss. And this is not for, we are trying to do it in one center with our, let's say, 10,000 cohort. But, you know, the best scenario would be if we could do it on 1 million cohort all around the country. So why not? But still, it costs money. And we try to convince many people, many that are really, let's say, decision makers, but still, this seems to be kind of long term, and the benefits are not short term. So this is why maybe we have some difficulties in convincing decision makers that that should be financed as soon as possible. And of course, somebody has to do it. It's about people. So we do everything to actually to educate these people. We don't have too many people that really can be at the same time, maybe not to be at the same time, but we don't have too many clinicians understanding the bioinformatics and too many bioinformaticians understanding the clinics. So we have to kind of, educate them, behave to bring them up. So what we are doing, we actually are trying to implement the quite, I hope, modern curriculums of our PhD programs. One of them is already ending, the other one has already been accepted by the European Commission, and we are the leader of Horizon 2020 program in the framework of co-fund actions. When we are right now recruited, we are in the process of recruiting of 15 international young researchers who will be actually on both sides, on the clinical side and on the bioinformatical side. That's a very challenging attitude. It will take us a lot of effort to make them to understand both sides, but I think we have to do it. Otherwise, two sides will be speaking two different languages and no one will understand what we are talking about. Thank you very much for your attention, and as I said, everybody is welcome to collaborate. It's been a pleasure to be here.
So a few questions, any comments?
I've already mentioned 3 times.
So yeah, I have to add it. It's not only our institute. It's a really over the 2 years I think we are collaborating. It's a really nice example of a collaboration between total scientists who are really thinking different way than clinicians. And I remember our first meetings that we really couldn't understand each other. And we have bioinformaticians as well who are really talking in a totally different language as well. But I think over those years we are talking more or less the same language now. It's really nice. And what is really important that we have this PET MRI that we, for example, are seeing some kind of correlations with transcriptomic data. This is really nice. That was never really studied. And what is also important that we are collecting other tumors, like ovarian cancer, for example, different tumors from different hospitals in Poland. So,
but in the same way, in the same
in the same way.
Yes.
So it's really the same way, and we have different things that we can study in the future. So
sure.
We are open for the cooperation.
Yeah, it's a job for the next decade at least. So we will see what's going to happen with arginase, whether it's really why it wasn't consistent in clinics yet.
Thanks. This was a great talk and a really interesting initiative. So I'm wondering, I think there may be a caveat to collecting everything from everything. Is that you...
Not collecting everything.
Gathering all the data from every sample, because then you end up with a database where there is relatively few samples. Like you said, samples are in the thousands, whereas the variables that you can consider for each clinical outcomes are in the, I don't know, tens of thousands or hundreds of thousands or even millions. Yeah. So how do you deal with that? I mean, I'm assuming the goal in the end is to use machine learning to generate hypotheses and to predict things. And if you have a data set where there is many more variables than there are samples, you're prone to overfitting, and you're getting hypotheses that look correct, but in the end they end up being incorrect.
Yeah, that's a very good point. So this is why what I was mentioning at the very beginning, before we get to this machine learning and this creating the hypothesis by some kind of let's say algorithm-based hypothesis, what we are first do, we will try to confront our data with something that is already known, with the known driving mutations, with the known therapies and some kind of the, let's say, the test and analysis that can predict the responsiveness to current therapies. So we don't want to start from nowhere. We at the beginning we'd like to start to stick to something that's known, and then step by step to see what's gonna happen. Otherwise, we're gonna get really synced if we in the crowd in this huge amount of data. So. But this data will stay there. In the course of time, we will be developing new tools. So then, and of course, we're not going to end with this. We just started, but our decision is to continue with hundreds of new patients. So for the purposes of this project, we were kind of obliged to do the 150 of operatable patients. And we altogether have like 500 lung cancer patients. So it's not a huge number. However, in the course of time, 2, 3, 4 years, it's going to cross the 1,000 numbers. So then, of course, we will be always looking at something. Our strategies go from the small to the bigger. So we're totally aware of this caveat, and we will be very careful looking at this. But as I said, we start from something that is at least partially known so far, and then we'll see to kind of go to these areas that are completely unknown. But we are realistic. We are idealistic on one side, but realistic on the other side.
Yes, a good point. I believe that the name of our session is molecular biology. So the question is how you can put molecular biology in your omic data base and regulations, because in general we are doing some biomarker studies and some omics in very tiny amount of people with very specific conditions like rare diseases, and at some point we are struggling with molecular biology because we can generate hypotheses, but later on you need to prove it, and to actually to answer the question, because as I saw, as you put in your presentation, that for instance you have a lot of variants in the genome, which can divide the people who are not responding to checkpoint inhibitors. Probably behind of this variant there are some really important ones which can be struggled.
It's not the specific variants.
I know.
Just the number of the.
It depends on the variability of the cells. If are more variable, immune system can recognize them and kill. So this is kind of surrogate of the really biology behind of this omics stuff.
Yeah, again, very good point. And I think what our data set it can be really useful for. So, for example, if Professor Młynarski is coming with some hypothesis that based on his existing experience, so he can get back to our, let's say, omic data, and then we can kind of direct the analysis strictly looking at this pathway. Otherwise, we can get lost.
I'm really invited. Thank you.
Yeah, sure. No, I'm here just to invite, not to give a talk. So I'm here to share the news and, of course, invite everybody on the board. But this is hypothesis-free approach. So we are not saying that several molecular patterns are better or worse or more interesting or not. I think that can work in the other way around. If somebody has some kind of hypothesis, it can come up to test this hypothesis even in silico. Looking at that, what's happened actually in our real world settings here. So I'm not saying that we, of course, as I said, answering the previous question, from the very beginning we will see how the existing knowledge can really work in our model with all this EGFR, ALK fusion, and BRAF, and so on mutations. How do they really work in our model? How do they can they really be used? Or maybe what are the other genomic patterns that can be related to them? Or what are the epigenomic patterns that can be related to that we already know, that has been checked in the clinical settings? So this is something which we'll start with. And then if somebody has some new hypothesis, so let's check it. It's difficult to do all the hypotheses in one, but we can do it the other way around. Some hypotheses can be tested just based in a very specific directed way using our data set. So again, welcome.
Okay. Thank you.
The last question.
Thank you, Professor, for a great presentation. I really appreciate the whole effort. I think that it's really a great direction, and it's worth of money. I just wanted for a while to refer to the question which was by the previous which was asked by the previous person. I wanted to just tell a little about different story. I personally deal with the corpus which have like 150 millions of real patients episodes. And this is, of course, very heterogeneous database, which is the composition of various subpopulations coming from various diseases and from various comorbidities, etc. However, I have to admit that still we deal with the similar problems, which is in fact the problem of let's say what's the minimum required sample size for some particular subgroup of patients. In other words, speaking, even having very heterogeneous, even having a big corpus, because of the heterogeneity, still in some subgroups we have similar problems, which are the sample sizes. At the same, what we noticed is that when we add when we have the additional context for the given patients, the quality of the patterns is increasing and the confidence is increasing. So from this perspective, looking at your project, where you have the really high quality data, when we can look at the patients from the various perspectives, right, for various omics perspectives, I believe that still with a lower number of supporting patients, supporting cases, still we can produce high quality patterns comparing to other databases where we do not have so many additional context spaces. So from this perspective, I have to say that really this is a great strategy that you focus on the, let's say, just registering those contexts for your patients, and as a derivative producing really high quality data. This is really good strategy, and I believe that this is only a question of time when you will have, let's say, a more representative corpus. At the same, controlling the, let's say, confidence factor, you can always say where you are, let's say, where your prediction is stable or not. So from this perspective, you will always stay in the safe zone. So again, I really congratulate your strategy, your direction. I believe that this is really very helpful project for many patients which are waiting for something like this. And I also believe, as a AI scientific guy, that this is something which are, let's say, producing many challenging, let's say, threats for many machine learning people, just to try, let's say, their techniques. So really very great work, and I'm crossing fingers, and yeah, that's all.
Thank you very much. Thank you.
Thank you.
Thank you. And of course, we are not ending. We will be increasing the group, and will be, of course, widening this. Yeah. Yeah. Sure. Thank you very much.
Thank you. Joanna Jurek. She's going to answer the question how a single nucleotide polymorphism may impact on post-translational modification of retinoid X receptor alpha through simulation.
So good afternoon. Thank you very much for the opportunity to present the work, which is part of my Bachelor of Science final year dissertation. And basically, I'm going to present that. And the topic which I focused on is about the nuclear receptors, and I mostly focus on the group retinoid X receptors. And basically, my work was changing over the work as normal. And the first aim of the study was basically to explore different isoforms of the retinoid X receptors and see what, because the challenge basically is that the retinoid X receptor alpha is the main isoform, which is the well-known one, and the other ones, which are the reason, which are created as a result of the splicing, are the beta and gamma. And my first aim was to explore whenever they are similar or maybe they are distinct. And then I focused more about the post-translational modifications, and I mostly focused on the simulation and basically the assembly of sumo protein assembly to the consensus motif. And whenever I was researching the literature, and basically I was trying to explore the background of the study, because it was my first topic when I focused on mostly on the molecular biology, I find out that this consensus motif is really, really important in terms of the simulation. And most of the studies said that, okay, the lysine is an acceptor site, so the sumo complex is attaching here. Of course, environment is also important, but there is no clear what should be around the lysine. Everyone was focusing on the lysine. But I was researching another source, which is exome variant database on the Washington server, and I find out that, okay, the one confirmed simulation site of the lysine, basically the environment of this lysine was modified, and the modification was present in the population. So I said, well, I will focus on that. And that's the study hypothesis, and this polymorphism was occurring around the sumo site, which is lysine 245, and the polymorphism present within the population on the server was exactly was a glutamic acid was exchanged to another lysine. So technically, there was disruption of the consensus motif in terms of the whole environment. Of course, the lysine was still, the acceptor site on lysine was still present, but we were talking about the environment of the lysine. And as I mentioned, I firstly focused on the computational part of it, and I was trying to explore, compare. I was trying to see whenever the possible polymorphins will be present within the other isoforms. Unfortunately, no. It was only specific for the isoform alpha. And then I moved to the molecular part of the study, which was basically designing the mutant on expression construct. And of course, I was planning to move into the sumo assays and reporter assays to see whenever it's affecting in a cell culture, but unfortunately projects at our university are only designed for maximum 5 months, including holidays, so there was no aim. So I thought that, well, achieving the stage of the study whenever I had the mutant created and was ready and waiting for the another study with into cell culture was possible later on. So in terms of the results, as I mentioned, I firstly focus on the comparison between the isoforms. What I find out was that the isoform alpha and gamma was the most similar. So I would say I can suspect that the function will be similar, but the polymorphism was not present in the gamma. Was only, as I said, was specific for alpha. But when the beta isoform was the most distinct one. Oops. And then I moved to pairwise comparison just to more focus on and see what the differences are. And I also used the SUMO predictors to see whenever my expectation where the possible SUMO sites can be within the sequence can be present. And what about in terms of the computational tools? And finally, this is the table which presents the mutation, the polymorphisms I found. And the thing was that not only isoform alpha has own polymorphisms present in a population, also beta and gamma, they are of course different. But I think that might be for us the indication that some population this results are from the African and European populations, so that can be for us an idea, which of course requires further studies to say, okay, whenever we have problem with assembly for SUMO modification to regulate the nuclear receptors, it might be because of the presence of the change polymorphism or maybe even mutation. My molecular work was was designed for the tissue culture also, but the timeline was really, really tight, so I only achieved the stage when I create the mutant. And I prepared the sequence just to confirm that okay, mutant was created, and just for own interest, I decided okay, because the polymorphism was about changing one glutamic acid to lysine, which is completely different sort of amino acid in terms of the charge. Probably the effect might be different, and that's just for own interest and just an idea to see this, and that's the difference. So we see that we have a big change. Maybe it's not a big, but we have a change in the isoelectric point. So it's something around there, but unfortunately my knowledge in terms of the protein, it was not as such, and I just want to mention for my own just to clarify that we submit the project because we must submit project with results about April, and this idea to study the protein was in May, so it was just like, okay, maybe we add a little bit and maybe we show something else. So this just was for own interest. And just in summary, as I mentioned, this presence of polymorphisms might be an kind of the indication that okay, here we can try to explore this area more, because this difference might be associated with different responses to the SUMO modification and other post-transitional modifications. But of course, it requires the further study. So thank you very much for your attention.
Thank you very much for your presentation. I know that this is premature, but we have time for a question, so not leaving.
No, because I was thinking that I need to pass to Marcin.
No, it's fine. It's fine. Any questions? I have a question regarding. Oh, it's okay. So later on, me.
So this is really interesting work. What do you think might be the effect of SUMOylation on the function of RXR isoforms?
Thank you for this question, because now I realize I forgot to mention that the sumo assembly of the sumo protein was associated in terms of the retinoid X receptor to the downregulation of the activation of the retinoid X receptor. So technically, my understanding, how I can possibly think about this result is like, okay, if we have something within the motif sequence which is affecting the sumo assembly, because I don't know what the effect will be because I haven't tested. So I presume that, let's say, that disruption of the motif will cause that our sumo assembly will be poorer. So if we have a poorer assembly of it, we will have a problem with the regulation, downregulation of the receptor, or with activation of the receptor, because retinoid alpha is mostly working within the heterodimer. In my understanding of, it's not permissive, so it needs the permission from another part of the dimer to say, okay, you can work or you can't. So I think it's not that it might cause the other version of the regulation of the sumo, but I don't know, because I don't know what the effect will be because I haven't tested, and that's why I said that we need to plan the sumo reporter assay, because then we will know, okay, if we have this polymorphism, this disruption, and we can compare, okay, the assembly is poorer or weaker, or it's more enhanced.
Right here. What is the native ligand for the RXR?
The ligands.
Yes.
Basically, it's a retinoid. Is a retinoic acid. That's what for retinoic X receptor.
For RXR?
Yes, that is just difference between I know it's a retinoid acid receptor also, but for RXR, I remember that was the synthetically prepared LG with numbers, and I know that the retinoid acid was also shown in the studies as a ligand.
Okay, I think it's worth to check it.
Yeah, I will. Thank you for your comment.
One question regarding the SNP, single nucleotide polymorphism you found. Is it possible to find this variation in the ClinVar database, and how it is annotated in this database, if it is?
I remember I was trying to find the effect because I found it that it's possibly damaging, but that was the information which comes back to me from the server. But I think that was something which caused me a problem to find. Like, that was not easy accessible, but maybe my skills in terms of searching that was just really at the early stage.
Okay.
They need improvement.
Okay, so thank you. Any other questions, comments? Thank you very much for your
Thank you very
presentation
much.
again.
Thank you.
And let me stop at this point and finish our session. I think it was somehow a provocative session, and also we have very good lectures and a presentation of original papers. And I think the further discussion will be just after the session. Thank you very much.